Swimming in hot water.
I'm the worst.
Is that right?
Yeah, that's good. Yeah, that was my face. Yes, so thank you very much. My name is Jackson Sage. I am a second year PhD student, and I'm going to give my talk, Swimming in Hot Water, Environmental Change from a Physical Perspective. Now, the organisms that I work on are phytoplankton. These microscopic plants are found the world over, from rivers to lakes to our oceans, from the North Pole to the equator to the South Pole. They really are a global group of organisms. The species that I work on is Tetraselmis zooxeca. So around about 10 to 12 micrometers long, so around about 1.5 times the size of a red blood cell. And it has these 4 flagella, which it uses to swim. And it swims very, very well. Here we can see the effect that temperature has on swimming speed of these cells. So at the left, at 10 degrees, they're buzzing along at a respectable 16 body lengths per second, so still quite quick. But on the far right, at 30 degrees, they really are flying. They're going around about 35, 36 body lengths per second. The thing is with these cells, most people expect, okay, there's a change in swimming speed with temperature. That's due to the effect, okay, we increase temperature, chemical reactions in the body begin to occur quicker, the cell's metabolic rate increases, therefore they can move their flagella faster and swim faster. That seems plausible. The thing is that when you are as small as a phytoplankton, you have to consider the effect that viscosity has on swimming speed. So just to clear up, viscosity is essentially the stickiness of a fluid, or the resistance of the fluid to flow. The key point that I want to make here is that the water that we're talking about is literally just seawater. There is nothing special about it. It's exactly the same water that you'll find a blue whale in or a tiny swimming sperm. The thing that makes the difference is the size, because depending on your size, viscosity will affect you in different ways. Now, we can kind of scale this up if we want to imagine, okay, how exactly would it feel to be a swimming phytoplankton, a tiny, tiny organism? And we can do it by using a quick equation, which I won't go into, but we can calculate that the viscosity of the fluid will be similar to this. So this is blackstrap molasses. Has a viscosity of around 62 million millipascals, about 60 million times stickier than water. If you could swim the width of an Olympic swimming pool, so about 25 meters, in a second through that fluid, then you would be able to swim as well as these phytoplankton can, which is just, I still get puzzled by it to this day, I'll be honest. The really interesting thing about viscosity, though, is that it is temperature dependent. So as we increase temperature, we see a decrease in viscosity. Obviously, for the inverse, decreased temperature, you get an inverse in viscosity. Now, if you want to test this out at home, get a bottle of vodka and put it in the freezer. The temperature of your freezer won't be cold enough to freeze it, but when you pour the vodka out, you'll notice it has a syrupy consistency. That's due to the effect that temperature has on viscosity. So try it when you get home for science reasons. But the interesting thing is that if you think back to the video I showed you with the different swimming speeds at different temperatures, is that change in swimming speed that we see, is that due to the effect that temperature has on metabolic rates, cells swim faster? Or is it due to the effect that when we increase temperature, viscosity decreases, the water becomes less sticky, and therefore the cells can move easier? And that was the first question I wanted to ask. Is the relationship between swimming speed and temperature due to physiological or mechanical factors? So, to answer this question, we initially set up a master culture of these Tetraselmis cells at 20 degrees for 20 weeks. The reason for this is I wanted to know exactly what kind of conditions the ancestors of the cells that I'm going to be working on were exposed to. If I just had the cells just popped on the lab bench and they were at a random temperature, whatever it was that day, I couldn't be certain if there was any pre-adaptation to a certain condition, so I wanted to make sure I controlled that. And plus, to be honest, the 20 weeks gave me a little bit of time to play. From that master culture, we divided it into 3 treatments: T20 at 20 degrees, T25 at 25 degrees. Note that the viscosity is less than it is at 20 degrees, as we would expect. And then we had this special treatment here, T25F, at 25 degrees, but with a viscosity of that 20 degrees. We did this by using an inert polymer called Phycol. The great thing about Phycol is that, when compared to other polymers that you can use, it only affects the viscosity of the fluid. It won't change the density, it doesn't alter anything else, it's just going to change the viscosity, which is what we wanted. All three of these treatments were cultured for 10 weeks, or around about 40 generations, and there were samples taken on a weekly basis. From each treatment, there were 5 replicate cultures divided up, and the velocity of 100 cells were recorded per treatment per week, so there really was a huge amount of data that went into this. And this is what we found. So blue at 20 degrees, red at 25 degrees, this green line in the middle, T25F, so 25 degrees but with a viscosity of 20 degrees. What we found was that for a 5-degree increase in temperature from T20 to T25, there was a 24.7% increase in swimming speed. But by using the T25F culture, we can figure out that 13.8% of that increase was due to an increase in metabolic rate, 10.9% due to a decrease in fluid viscosity. So that's something that's been done on larger organisms, but it's not been done on phytoplankton before. So from the first question, we can say that physiological and mechanical factors contribute almost equally to an increase we see in swimming speed. It's about a 56 to 44 ratio. And metabolic rate is not the whole story when you're as small as a phytoplankton. So you've seen that this experiment was carried out for 10 weeks, a number of generations, so obviously we were interested in change over time. So that was the second question I had. Can my cells acclimate, which is within an individual, or adapt across generations to an increase in temperature or viscosity over time? So you can tell I love my flow diagrams. Everything above here is exactly the same as you saw before. But what we did is we had this thing which I call weekly ancestral resuspension. That was done at the same time. So on a weekly basis, when I was taking my normal samples, I'd take a little bit extra, filter it from its environment, and then resuspend it at 20 degrees. The whole purpose for this was, let's say for example, my cells that I've got at 25 degrees. They've been in there for, say, 6 weeks. Does being exposed at 25 degrees for 6 weeks give them some kind of advantage when exposed at 20 degrees than those who have been exposed at 20 degrees all along? Does that exposure to increased temperature give them some kind of benefit or loss? So just to clarify, the top line again is T25. This is resuspended T25 cells. This is resuspended T20 cells. Obviously we resuspended the T20 cells as well, just for control, due to the effect that you can have on filtration. You'll note that there is still a small difference in swimming speed, but it was not significant. There was not a significant difference in swimming velocity between T20 cells and resuspended T25 cells. However, for T25F cells, what we found was that there was an increase in swimming velocity over the course of 10 weeks. So week on week on week, but up until week 6, there was an increase in swimming speed, up to about 18% increase in swimming speed. And that was completely unexpected. We were, it still puzzles us a little bit now, in fairness. So the interesting thing with these guys is that the lifespan of the cells we're talking about is 7 to 10 days. This change that we're seeing is occurring over 6 weeks. So something is happening from generation to generation. There's a gradual increase. So that was what we found out here, that there was an increase in swimming speed over the course of 10 weeks, 18%. Okay, how long is it going to take to get rid of that? It's taken 10 weeks for them to gain it, how long does it take to disappear? And that's where this little bit on the end came along, reverse acclimation. At week 10, we filtered the cells from their environment, resuspended them at 20 degrees, exactly the same as before, but then cultured them for 4 weeks, so we could see how long it would take to disappear. The sampling rate on this one, we took samples on a daily basis for the first 2 weeks. The whole purpose for doing, swapping the daily samples is, you think prior to this there was 10 weeks of experiments, and then prior to that there was 20. I didn't want to miss out by a day after 30 weeks. So that was the reason for that. And what we found was that T25F cells retain their faster swimming speed here for 4 days, or around about 3 generations, before becoming non-significantly different from the other treatments. So as I can't remember who it was that said it during the talk the other week, but it kind of bolsters what that person said, is that it's much easier to lose something you've gained than gain something that you don't have in the first place. So that kind of stuck with me about that. So to answer that second question, adaptation to increase viscosity is possible, but it takes about 10 generations. And the mechanisms that Tetrasalmus employ in its adaptation have yet to be confirmed. So that's where we're up to now. How is this occurring, and how can we find out? I was quite lucky a year ago or so, where I was able to borrow a high-speed camera from the engineering instrument pool. And that was our first step to repeat the experiment, annoyingly, but look at it in high speed, which was great. So both of these videos are recorded at 3,000 frames per second, which is the one on the left that I'm interested in. So what we did is we looked at body length, to flagella length ratio, swimming speed, beat frequency, distance travel per beat, all of this information across the course of 8 weeks, just so we can see if there is some kind of change between treatment and how these guys are achieving their faster swimming speed. Is there, over week on week, a change in, say, for example, body length to flagella length ratio? It's not a spoiler there or anything like that, that's so far. Take what you will from that. So that was the first thing. The next thing that I'm interested in looking at is if there's a change in flagella morphology. So this is my Tetrasalmus cell. It's full flagella, and it's got these tiny vestiginemes, these hairs, which stick off the end of the flagella. There's been studies on a different type of phytoplankton called Chlamydomonas, which is a very similar size, freshwater phytoplankton, and they found that cells which are bold, which don't have these hairs, can move their flagella around about 10% faster than those with vestiginemes. The thing is that they also swim about 20 to 30 times slower than those with. So without vestiginemes, you find that you're able to move slower through the water. Best way I can explain that to you as to how it happens. Imagine you're in a rowing boat, but you're using your oars the wrong way around. You probably move those oars quicker through the water, but you're not going to go anywhere. So what we're going to be doing is using either AFM or SEM to have a look at these vestiginemes. Vestigineme length, vestigineme frequency, distance between them, see if there's any change between treatments over time. That could possibly be the answer to our question, obviously in these different speeds over time. So we've gone from looking at whole cells, down to how the flagella move, down to the flagella morphology. The next thing that I'm going to be working on with Dr. James Flint is going beneath that and looking at the flagella substructure. So all eukaryotic flagella have this same kind of setup with two central microtubules, these 9 outer microtubule doublets around the outside, and these motor proteins which they use to move the flagella. We're going to be doing some molecular biology, doing some sequencing work on the samples, and seeing if there's a change in the proteins that have been expressed between treatments. This is something that I've not worked on before, but it's something that I'm really sort of quite eager to sink my teeth into. So to conclude, physiological and mechanical factors are almost equally responsible for changes in swimming velocity or temperature. Tetrasalmus can adapt to increase viscosity, but the adaptation appears to be linked to kinematic or morphometric compensation, but we are not entirely certain at the moment. So with that, I would just like to thank Professor Stuart Humphreys, Dr. Domino Joyce and Dr. James Flint, who are my supervisors, all of the Physical Ecology Lab, NERC, who are main funders for my PhD, University of Lincoln, University of Hull, EPSRC, who funded me, and Assemble, who were part of a EU grant I was awarded, who gave me some nice different phytoplankton species to play with under the high-speed camera. Thank you very much.
Can we have at least one light back on something?
That's all right.
So, you don't want to. Mate? Maybe not that one. That's better. So now we're going to the effect of light and photo adoption. Anybody got any questions, Daniel?
The explanation you gave is the classic explanation also given for the fecal response, that by raising temperature you can suppress the iron more effectively and therefore bacteria are less likely to be produced. Has anybody looked at whether or not that sort of change in temperature makes polymorph nucleosides more easily, affects them more than the bacteria they're likely to be involved in?
I have no idea. I'm afraid it's this.
There's some indication that clearance rates in airways increase with temperature, and so there's the kind of idea that you increase temperature to kill the bacteria, but there's also a small indication that you might be increasing the rates of ciliary movements, I think exactly the same things for Gemma, to remove particles from your airway, and that might be another secondary benefit to fecal response.
The actual motility of the body of the defense cells versus
They
bacteria.
could speed up, yeah. I mean,
Shapes.
I think, yeah, no one's really looked at it, but there could well be a link there with the movement. But bacteria are affected in essentially the same way, unfortunately.
It's different, probably they change their shape from the mentioned effect.
There could be, but yeah, it's not anything that we've already looked
So, I'm sorry if I missed this, but what is the, you're proposing an adaptation which implies a selection pressure, which would suggest to me that, are they competing for food in the fastest?
So essentially the reason why they are trying to swim faster, you asking about why?
Well, they're not trying, but
Well, yeah, sorry,
the
yeah.
ones that swim faster are more successful, is what you're suggesting. So what's driving that? Are they, is it food limited in there, or why would there be selection? I'm saying in your lovely cultures where they
Yeah.
live a lovely life.
So I'm not necessarily saying that it is that faster swimming speed is beneficial. It might not be. It might just be. Remember, these are just, although we're watching swimming phytoplankton, there isn't as much of a sort of cognitive effort to gain particular food, for example. It could be possible that they're just swimming faster as a response, that it is just, how can I explain it? That it might not necessarily be beneficial for them. So one thing that I wanted to look at was seeing how chlorophyll A densities change between these treatments. And say, for example, okay, these guys are swimming faster, but using chlorophyll A as a proxy for health, if that changes. So I'm not entirely sure how well that answers your question, I'm afraid. But
I guess I am, still a bit puzzled about what you think is what you called it an adaptation. That implies to me selection.
yes.
So what's driving that selection? Or did I miss something there?
I mean, you can have, say, for example, it could be food acquisition or nutrient acquisition.
Yeah.
Quite possibly. And these guys, obviously, they, rather than other phytoplankton, they do use motility as a source for either gaining nutrition or gaining light. But as to which selection pressure it is, because all of those were kept uniform across, it's difficult. My point is that the change in swimming speed, because we are seeing it across generations, that's my argument for calling it an adaptation, because we are seeing a gradual change over time, which is not from the same individual. If it was from the same individual, we'd call it acclimation.
Yes, absolutely.
But in terms of the pressures, what are going on, I'm not entirely certain. Yeah, I mean, the main way we started off this experiment was to kind of look at just decoupling the physics from the biological side. So So I mean, that wasn't really the way we were trying to go with it in the first place. So,
Okay.
but it is something to
Really interesting, anyway.
And then this way.
Bit of a technical question. Where are you actually growing them? Are they in tanks? Are they in cultures?
So these were all set up in culture flasks. Culture flasks were popped into water baths at a given temperature, depending on what it would be. So we had 5, just basically culture flasks of them, which were aerated, triple filtered with air because we have problems with not infections,
Contamination.
yes, thank you, contamination. So yeah, and under a 24-hour light cycle as well.
Have you considered building up toxins? Because in effect you're growing batch culture. So you've got no influx of new nutrients, no removal of waste.
The cultures are refreshed on a weekly basis. So when I take my sub-samples, obviously any toxic that would be in there, obviously I'm not going to get rid of it, it's just going to be a dilution effect, and there's still going to be some in there. However, if you were referring to why the T25F cells were swimming faster, if you're saying that there was a toxin and if that was explained some way that we saw the result we did, I'm not sure that my cells would swim faster over time when they're being resuspended. And besides, these resuspended ones were resuspended into fresh cultured water. They were taken out and resuspended.
Just from my background of steady-state hemostat culture, where you have then a continuous level of toxins and fresh nutrients, what I was noticing is there's quite a lot of fluctuation on your points.
Yes.
I'm just wondering whether trying to create a continuous 10-week culture may actually eliminate some of those.
I mean, so we are getting fluctuation on the points. But when you look at the actual difference in side on, so we're going from, say for example, do you mean within culture? There isn't a huge amount there, but yes, there is some fluctuation. So
I'm just looking at those and seeing steady-state hemostat cultures, but you're kind of doing
different,
steady batch,
yes.
batch fed.
Yeah.
So might be an interesting way to look.
Definitely, yeah.
Yeah, lots of questions. Next one.
How do the algae swim in such a viscous fluid? Like, how do they swim so fast?
So when you're in a viscous environment, luckily I have something prepared, which is always good. Best way to think about it is using a secret. Surprisingly. So if we imagine
Best way to
this...
think about everything, Jack. It's the best way to think about everything.
Yes. So imagine this is an algae cell, and these, rather than its flippers, are its flagella. It's doing this steady beat movement. In water, it's doing the same thing. You've got this thing called lift-based thrust, where basically it's pushing the fluid behind it and it's moving into the space that it's created. That's how we swim in water. When you are a phytoplankton, sorry, you're not moving the fluid out of the way and moving into the space created by it. You're essentially gaining purchase on the fluid. It's a completely different way of thinking about it. So as you move, as it moves its flagella forward, it almost grips onto the fluid and then drags the body with it. So it's a completely different way of trying to think about it, which is what makes it quite cool, working at this small scale.
In the interest of fairness, 2 more questions and we're going to stop. Otherwise we'll be here all day.
This is a really stupid question, but how do these guys reproduce?
Not stupid, no, because you do get, with phytoplankton, you do get some which sexually reproduce and some which are asexual. These guys reproduce through binary fission. So they're just dividing cultures. That's dividing cells, sorry.
So it's just-
Maybe not with the kind of sound effect. Maybe someone in bioacoustics.
So I just want to go back to the point about what the pressure could be there. It's just, so you're saying these guys, they react differently to say like, so like, do they go towards or they just go in random directions and hope that they bump into them?
So that's something that we were trying to look at, is sort of look at chemotaxis within the cell. There was sort of an undergraduate project we tried in there. The thing is that depending on whether you go to food or not, depends on your size. So there's something you can use called the Peclet number. There's a famous hypothesis. Vogel? Is it Vogel? Or I think of Reynolds, sorry. Called the lazy cow hypothesis, which basically says that with some of these cells, they essentially just it's the same as eating a patch of grass and then just waiting for it to regrow due to the effect of diffusion. So for some of these cells, they'll basically just diffuse, not move, and just wait for more to come their way on the conveyor belt. Whereas these guys, obviously actively swimming, it's going to be much more, they move and they rotate for nutrient acquisition as one of the things, as well as light acquisition as well, obviously it's very important.
Next question, Sheila?
Yeah, I can't remember what the question was. Oh yeah, so when you use subculture every week, did you subculture the entire population or just a sample of it and things when you...
So, let's use this. So this is my culture. Every week I take off 50% of the culture, from which I will do my experiments. And then I top up the rest with fresh media. So it's a 50% fresh change. So there was new nutrients going in on a weekly basis. There wasn't a reliable way I could find to measure the nutrient content in there, but obviously with, I'm sure I could have used your expertise when I was doing this, you would have told me. So that's how it was, how we were doing it. We were basically taking off half the culture, and then that was my lot to play with from each subculture.
So the answer is probably not then, but I was just wondering whether you were basically being the selection predator, and maybe you were actively selecting the faster swimmers by the way you were subculture. I'm thinking that due to the density at which these cell cultures were at, and the fact that it was, it's possible. It's not something that I've given much thought to, but it's definitely worth thinking about. Yeah, with me taking away half of the culture each week, if that's going to change. We did find that we did find that cell density had no effect on swimming speed. That was one of the things that I factored into my model. So I'm not sure how well that would work. You know, obviously you might expect, okay, that if there's less cells within the culture, less things for them to bump into, they may be able to gain higher speeds, but we weren't finding that.
Okay, so we thank Jackson for a very... I have mentioned this